Neurons are the cells responsible for complex mammalian processes such as long-term memory storage. In the past, single-cell RNA sequencing has had success within cell bodies, but there is much to discover in distal regions of the cell. Using RNA sequencing, we can explore how genes code for proteins that contribute to the performance of highly evolved tasks. Specifically, it can clarify neural development, plasticity, and regeneration. After mRNA is sequenced, we can use these sequences to explore regulation and the molecular diversity in neurons. It's hard to access neurons of mammals, especially humans, so scientists use methods like assessment of ex vivo neurons or pluripotent stem cells. These stem cells can develop into any part of the embryo, and thus every human cell, including neurons. To dive deeper, we will explore the axon subtranscriptome, which is sequenced using growth cone sorting RNA sequencing. But first, how are sequences localized in the axon? A wide range of proteins, such as motor proteins, associate with mRNA transcripts and carry them into storage. A cell then receives a signal to start translating in many ways, but one example is receptor-ribosome coupling. It activates translation and forms many axonal proteins. It's important to consider how this gene expression is regulated. One factor is the quantity of ribosomes. Another factor, which was discovered as a derivative of RNA sequencing, is initiator upstream open reading frames. They may have repressors bind and halt translation when proteins aren't needed. What is the significance of these upstream open reading frames? All vertebrates use them as a means of translational regulation, including 62% of the genes in zebrafish. In conclusion, RNA sequencing offers special insight in hard-to-reach areas of neurons. It informs us on regulatory processes and specific protein products and functions. It's important to realize that proteins may be localized, like axonal proteins, or may move around the neuron, like motor proteins. They can help us understand how the brain can do such complex things and ultimately what makes us human.